I'm sorry, I don't think we're in the original schedule, so a bit of a mix-up in who's going next here. My name is David Reed, and I work for the Bionetics Corporation at Kennedy Space Center. Next slide, please. We work in this building that was built shiny brand new just 6 years ago. I think we moved in, called the Space Life Sciences Lab. And really, we have three things that we do there on the Life Sciences Services contract. The first is we provide launch site pre-flight processing laboratories, and you're going to hear a lot more about that tomorrow when the launch site people talk about the process of managing your payload and getting into a laboratory for on-site processing. But the folks on our contract are the ones that actually implement that outfit the labs and make that possible for you. But we're mostly here to talk today about implementation partner kind of work. And in that regard, we do a lot of the same kind of things that you've been hearing. We have developed an extensive suite of hardware over the course of about 20 years and certified it all for flight. And in addition to that, we have a strong background in the integration aspect of it, doing that mountain of paperwork that you keep hearing a recurring theme about over and over again. Next slide, please. Gosh, if I knew that the last folks' presentation was going to look like this, I probably would have skipped this chart. But the take-home message here, which is bottom line, is that we've built a lot of hardware, and so a lot of it has flown repeatedly in space over time. Our hardware has been predominantly focused on plant systems and non-human, non-vertebrate biology, although we have some experience doing integration work with some foreign partners with some aquatic systems. We'll talk about that in a minute. Next slide, please. The ABRS is kind of our flagship piece of hardware right now. It was originally designed to be a plant growth box. It has two completely independent volumes inside of it, but we really like to sell it to you today more as a controlled environmental chamber box. It does more than just incubate, maintain temperature. It also controls a lot of other environmental set points that are important to conducting biological investigations. Things like maintaining a constant carbon dioxide level. If you're not aware of it already, that fluctuates quite a bit at times on the space vehicles, and depending on the sensitivity of your system, that can confound your results if you don't keep a constant level. In addition, it has an onboard scrubbing capability for scrubbing out things like volatile organic compounds or other selective compounds. We can load it with media of your choice in order to perform that function. Additionally, if you are conducting a plant experiment, it has closed loop recirculation of condensate back to what would be effectively the root zone of your plants. It's a hybrid. It can fly. Although it's currently on the ISS, we have a second unit on the ground here, and there's talk of potentially building a third. So it can both traffic up and down in the shuttle and also operate as an express rack, sub-rack payload. Next slide, please. This is just kind of a cutaway showing the inside of it. As I mentioned, on the left there, you see two large volumes and that kind of half pulled out box. You can think of that as, well, if you're growing plants, you can think of that as the pot that the plants would grow in, and they grow up towards that kind of pinkish lighting system at the top. That lighting system is selectable in terms of, although it's solid state based, we have a number of different color LEDs in there. We can optimize for plant growth. We can also provide a white light response so that you can, we have onboard cameras there that allow you to take images of what's going on. But more importantly, this environmental chamber has essentially a plug in it, and that plug allows us to plug in a number of other pieces of what we call experiment unique equipment. The piece of equipment you see on the right-hand side there is a GFP imager that was designed to do real-time non-destructive imaging of some Arabidopsis plants, but it provides both power and data transfer to the ABRS, which then pumps all that information down to the ground. So within this controlled environmental system, if what you need is a constant environment to conduct your biology, it's a relatively straightforward task to develop a smaller piece of EUE and even other implementation partners' pieces of EUE to place inside. Next chart, please. The biotube we put here, although it's a bit of an old piece of hardware, it was originally designed for flight on the shuttle. It points to kind of an interesting aspect of spaceflight and a kind of different approach that we've taken. Anytime you're using fixative materials that we talked about, or other so-called toxic materials. And incidentally, a spaceflight definition of toxic is probably different than your laboratory definition of toxic. Even water in certain volumes becomes a different tox level, which may be counterintuitive, but that's the way they look at it. So what we decided here was, instead of engineering a lot of small systems, each individually with redundant levels of containment, which is fairly costly and bulky, what we have is, in this picture you see a large, looks like an inverted blue cake pan in the back. That big cake pan itself provides 3 levels of containment at all times, so that the subsystems inside of it are relatively simple and only provide single levels of containment for things like glutaraldehyde or formaldehyde. So that way, if there is a leak, not that we're expecting there to be one, it's contained within the much larger volume, as opposed to having to have 3 levels of containment on every line, fitting, and subsystem. Next chart, please. I'm going to show you a number of pieces of equipment that we call BRIC, not to be confused with the ice bricks that you saw before, but the biological research in canisters is kind of the theory here is when you can, keep things simple, because the simpler a piece of equipment is, the much easier it is to fly. As you look at this thing, it looks like a roughly rectangular brick, and it's engineered around a form factor that a lot of investigators are used to, which is a petri dish. So this petri dish can be placed inside of this device that provides 3 levels of containment. You can grow either cultures or plants for some period of time, and then you can actually provide a chemical fixation. This device can be powered or not, and it's very small. Small is good oftentimes when you're flying, because they can find small places to put you where they might not be able to find a place to put a full-size payload or a rack-size payload to fly up and down. Next slide, please. Here's a different form factor for a brick. This uses the off-the-shelf OptiCells that you've seen in a couple of other folks' presentations. OptiCells were originally designed to do like mammalian cell culture work, but we actually were able to use them for a bacterial experiment that was conducted inside of a BRICS. Again, very simple. These things are passive. In the case in which they were used before, they flew up, and after some period of time, they were simply placed into that Melfi freezer that you heard about, and then eventually trafficked back to the ground. Very low resource required in order to conduct a very interesting experiment. Actually, the investigator for this experiment's in the room. I'm going to call him out. He's in the blue shirt back there. Raise your hand, Mike, so people can come badger you later about, no, raise it higher than that. Okay. So people can come ask you later about your experience with the BRIC Opti. Next slide, please. The BRIC-60 is a more cylindrical brick canister. Again, it was engineered around a stack of 60 millimeter petri dishes, but in the past we've also found all kinds of other things we can put in it. Teflon tubes, some of which had seeds, for example, rolled up inside of pieces of filter paper. Basically, you can think of it as an empty slate into which you can design just about anything. But the take-home points here is that, again, they're very simple. Almost all of our hardware is designed that it can be frozen and brought back. Bringing back samples in the case of biology is often a kind of a critical path item to consider. A lot of things either have to be brought back frozen or they have to be brought back chemically fixed. Next slide, please. BRIC 100s, again, same kind of thing, except engineered around 100 millimeter petri dishes. Some of these can be have a modified atmosphere placed inside the canister if you so desire. Next slide, please. The KFT, or Kennedy Space Center Fixation Tube, although it superficially looks kind of like the FPA that you saw in the BioServer presentation, it's in fact a bit different in the way in which it's used. If you see there's kind of an obvious break there between the actuator and the main tube. In a shirt sleeve environment, you can take your biology from whatever other system or subsystem you have grown it up in and place it inside of this tube manually, and in doing so you have a great deal of flexibility in what pieces or parts of that biology you place into the tube. Then you close the tube up and actuate it, and then and in the process of doing that, you flood the compartment with the biology with one of a variety of fixatives. There's a list on the chart here of fixatives that we've currently used in the KFT. We've got some others. There's a methanol, ethanol base that's being explored right now for a JAXA experiment. The key thing here is that this can be used, like I said, out in the cabin in the shirt sleeve environment. You don't need to be conducting this fixation inside of, say, the glove box. The glove box is a tremendous piece of equipment, but you've got to wait in line to use it. So this way, this is something that you can do anywhere in the vehicle. Next slide, please. As far as integration operations goes, well, you've heard the recurring theme here of folks that have been here, done it all, done it for a long time. We're another one of those folks. We have a lot of experience. You can see we've flown on pretty much every platform there's been. We're looking forward to flying on platforms that will be, such as the Dragon. We've had extensive collaborations with other space centers and other space agencies. So if there's a piece of equipment that we don't even necessarily own, we may have access to it, and we probably have experience integrating it. For example, there was an aquatic facility owned by the Canadian Space Agency that we integrated for them and flew before. Another aquatic facility owned by DLR that we have integrated and flown before. We do it all from design to development to integration, verification. Two things worth mentioning: we have something that we call our experiments monitoring area. You've heard people refer to as a payload operations center, or think of it as your own personal mission control. For us, it's a multi-purpose room in the Kennedy Space Center in our Space Life Sciences lab, where we traffic information both from our experiments on orbit and out to remote PI users. In addition to that, we have something that we call our orbital environmental simulator in the very next room. It looks for all the world like a walk-in growth chamber, but in fact what we do is we duplicate the on-orbit environment on either the shuttle or the space station by pulling down environmental parameter data, those that are valid and important to a biological experiment, principally temperature, relative humidity, and the relative concentration of carbon dioxide in the atmosphere on the vehicle. And then we have a control system that replays that to reproduce that varying set point in time in the orbital environmental simulator. We can do a completely synchronous control, we can do an asynchronous control, we can rerun your experiment multiple times as a ground control if need be. Next chart, please. Just myself as a point of contact. We've got a NASA point of contact here as well. These charts are not on your disk, but they will be posted to the website later on. Next chart. And I know there's a lot of acronyms that I didn't bother to define to you, but here they are for future consumption. Thank you.